Hello everyone and welcome to ScienceGem, your go-to channel for clear step-by-step walkthroughs of GCSE science exam papers. Whether you're aiming for top grades or just trying to boost your confidence, I'm here to help you break down the questions, understand the mark schemes and learn how to answer like a pro. So let's get into it. Today's walkthrough paper is GCSE Combined Science Trilogy, Higher Tier paper, Biology paper 2, and this is 2020. So this question is about DNA and genes. Which diagram represents a DNA molecule? Tick one box. So from your studies, you need to know that the DNA molecule is like a twisted ladder. Okay, it's a double helix. So obviously that's a hexagon. That's a fat molecule. So fatty acids, 3 fatty acids and glycerol. So concentrate on that one. That's glucose. So this is the answer. Describe the structure of a DNA molecule. So I've already given the answer already to this one. It's a double helix. You can also say that the DNA molecule is made up of 2 strands that are coiled or 2 strands that are twisted, and you can also just write the word polymer. You would still get the 1 mark. A gene is a small section of DNA on a chromosome. Complete the sentences. A gene codes for a particular sequence of amino acids. This sequence makes a specific protein. What is meant by the term genome? Genome is your entire genetic material. So, all the genetic material of an organism, but you don't need to say that, you can just say all the genetic material. The complete human genome is now known. Which important scientific advance was made using knowledge of the human genome? Discovering antibiotic resistant bacteria? No. Finding more foods to eat from tropical forests? No. Working out when the last ice age ended? No. It's tracing how Aboriginal people spread across Australia. So it's given us more information about human migration. A student found 6 different snails of 1 species in his garden. All the snails are different. What scientific term describes differences in characteristics between individuals of a species? The answer is variation. A change in DNA has caused snail P to be very different from the other 5 snails. Suggest why there might be an increasing number of snails similar to snail P in each future generation. So you can see P has a larger shell. So we say P has a larger shell. So remember the question is saying it's just why there might be an increasing number of snails similar to snail P in each future generation. So we look at P and we identify what this feature is. And the feature is, you can either say stronger shell or a larger shell. So what? So you need to then say this snail is more likely to survive. It's only 2 marks, so we don't need to go into too much depth. Remember, this answer is based around natural selection. So we say more likely to survive. and breed or reproduce. And that is it. Or you can also say that P is better camouflaged, so that means it's less likely to be seen and eaten, and so has a greater chance of breeding, or it will breed more. Okay, so question 2. This question is your nervous system. Okay. So human reactions are a response to an external change. Reflex actions help to protect the body against damage. Figure 2 shows the nervous pathway for a reflex action. A stimulus from the hot pan will cause the muscle in the arm to contract and move the finger away. So stimulus is a change in the environment or the surroundings. Okay. Describe how the stimulus from the hot pan reaches the muscle in the arm. So how is it possible that when you touch a hot pan, the muscle in your arm contracts, pulls your hand away? What's happening here? So what you see here is a pathway taken by a reflex action. So this is your reflex arc or reflex pathway. So what we say, so we start our answer off by saying that the stimulus is detected by the receptor cells, which are obviously in the skin. So stimulus is detected by the receptor cells, and then you say an electrical impulse is generated, which then travels along the neurons, and then you need to name the neurons in the order in which they appear. So remember, sensory neuron first, then it's your relay. Relay is always located between the sensory and the motor, and M for motor, and the way to remember that is M muscle connected to the M motor neuron. So an electrical impulse travels through the sensory neuron, relay neuron, and then the motor neuron. So far, I should get 3 marks. So this bit here, that's 1 mark. Electrical impulse, 2nd mark. Travels through the sensory relay motor, that's my 3rd mark. And then there's a 4th mark. And for the 4th mark, you can say that they are synapses. So remember the small gap that you see between neurons. Neurons never touch each other. You see a small gap, which is a synapse. Okay, so you can say the electrical impulse crosses the synapse. And you can leave it there, or because I've got my 4 marks, or you can then carry on and say at the synapse a neurotransmitter is released. But you don't need to go into details about, how the neurotransmitter diffuses across the synapse, because the question is just saying, how does a stimulus pass from the hot pan to the muscle in the arm? And you leave it there, you should get your 4 marks. A student investigated whether using the right hand or the left hand had an effect on the reaction time. The student only tested right-handed people. Describe a method for the student's investigation. So this is your reaction time required practical. It's 4 marks, and it says include the details of the test you would use for reaction time. Okay, so let's say how many people I will need for this investigation. So the student only tested right-handed people. So select at least 3 to 5 people, let's say. And then you could say, do the reaction time test. At least 3 times using the right hand. Okay, and then we need to describe what test. So we use the ruler drop test. Okay, so just a brief, because it's 4 marks, it's not 6 marks. So very briefly, just mention how the the reaction time test is carried out. So one student drops the ruler and the other student catches the ruler. Where the student grabs hold of the ruler with their thumb and first finger, that reading is recorded. And then mention that take several readings. So if you remember from the required practicals, it's at least 10. So take several readings times 10. So 10 readings basically. Then calculate a mean. And then you must mention repeat for each person. For the left hander now. And that should be enough to get 4 marks. And the other thing you can just mention that the people that we're using should be of the same gender, same age. These are your control variables. And like I said, that should be sufficient for 4 marks. A different student carried out an investigation to see if playing tennis improved reaction time. The student used 2 groups of 6 people. Calculate mean value X in table 1. So we're looking at all of these values, add them all up. And you've got 6 readings, divide that by 6. That should give you 1.8 over 6. But then your calculator, you should get 0.3. What is the dependent variable in the student's investigation? Dependent variable is what you measure. So what is the student measuring and recording? Reaction time. The student concluded, playing tennis improves reaction time. So our mean is 0.3 there. Give 1 piece of evidence which supports the conclusion. Well, you can see that students who play tennis have a shorter reaction time or a faster reaction time. So if you're reacting very, very quickly, your reaction time would be less. So look at this one, 0.3, 0.6. If you play tennis, 0.4, 0.5. So this is slightly higher than these numbers there. So you say students who play tennis had shorter reaction times. Give one piece of evidence which does not support the conclusion. So let's have a look. First of all, they've only used 6 people for this investigation, so the sample size is small. Okay, if the sample size was larger, we probably could have enough data to support the conclusion, but we don't in this case. You can also say that students who did not play tennis, so look at this one here, who did not play tennis, okay, has a shorter reaction time than someone who did play tennis. You can also say that there's a small difference between reaction time. Look, there's a difference of 0.1. 0.1. So the difference is very small. You can also say that there's overlap in times between the two groups. But the answer so when I'm looking at this data, the first thing that came to my head was, right, hang on, 6 people have been used. So I'm just going to stick to the first thing that came to my head, which was small sample was used. Or you can mention what I've mentioned earlier on. Question 3. There is a need to grow enough food for all the people in the world. So this question here, it's for the last section of the biology paper 2 spec. It's ecology. So what is meant by the term biodiversity? Diversity means variety. Bio, living things. So the variety of all the different species on Earth. That is a definition for biodiversity. So the variety of all the different species on Earth. Peat bogs are being destroyed because peat is used by humans. Destroying peat bogs also reduces biodiversity. Give one use of peat taken from peat bogs. Well, it's used as a fuel. 1 mark. Or you can say it's used as compost or fertilizer. Question 3.3. Explain why the use of peat is harmful to the environment. Do not refer to biodiversity. Now here I've said peat is used as a fuel. So if I'm burning peat, I'm releasing carbon dioxide, which is contributing to global warming. That's 2 marks. So burning releases carbon dioxide, which, second bullet point, which is increasing or contributing to global warming. Intensive farming, where a single crop is grown on a large area of land year after year, is more cost effective for farmers. It is efficient to use fertilizers and to burn the waste so the land is ready to use the next year. Planting a single crop reduces biodiversity. Suggest 2 ways that this type of farming can damage the environment. Do not refer to biodiversity in your answer. So let's start off with the burning. So burning crop, your plants. So burning crop waste produces CO2. You can also say, so it says there's just two ways that this type of farming can damage the environment. So you can say leads to soil erosion. What else could you say? You can say that, so if we're growing the same crop year on year, that would take minerals from the soil when the crops are removed. Farmers are using fertilizers, and we know that fertilizers pollute rivers, lakes and streams. You can also mention that using large farm machinery causes soil compaction. And you can also mention that when farmers use large farm machinery, that machinery will release carbon dioxide. Next question. Most of the world's production of antibiotics is used in farming. Antibiotics are given to animals to prevent the spread of disease. Remember, antibiotics kill bacteria. Explain how the widespread use of antibiotics in farming is a threat to human life. So, we can say that the antibiotic, obviously the antibiotic is given to the animals. The antibiotic will end up in the animal feces, so poo, which will flow into rivers, lakes or streams. So any bacteria, so remember now the antibiotic is in the rivers. In the river, you have bacteria in water. So bacteria in water or land, if the animal feces or urine flows onto land, that bacteria now becomes antibiotic resistant. Some of these bacteria in the water are human pathogens. And these pathogens or these bacteria will cause diseases which cannot be cured because they are antibiotic resistant, because the bacteria are antibiotic resistant. These pathogens cause diseases which cannot be cured because they are antibiotic resistant. So quite a challenging question. So question 4.1. In sexual reproduction, cells divide by meiosis to form gametes. Gametes are sex cells. So let's just do a quick recap. So sex cells, you've got sperm cells, you've got egg cells. Sperm cells are made in the testes, egg cells are made in the ovaries. So meiosis happens in the testes, meiosis happens in the ovaries. Which two statements are true for cell division by meiosis? So meiosis, remember you have your parent cell, DNA duplicates, so 46 chromosomes, Now you end up with 92. This cell divides, 46 chromosomes, 46 chromosomes, and then each of these daughter cells divide again. 23 chromosomes, 23 chromosomes, 23 chromosomes, 23 chromosomes. So in meiosis, 4 daughter cells are produced. They are not identical. They are not genetically identical. They are all genetically different. These are all haploid cells because they have half the number of chromosomes of the original parent cell. So 23, 23, 23. These 4 daughter cells, so if these 4 daughter cells were produced in the testes, these 4 daughter cells would be sperm cells. If these 4 daughter cells were produced in the ovaries, they would be 4 egg cells. Now, this is just DNA duplication. Okay, we're doubling here. Now, if you have a look, how many cell divisions can you see? There's division 1. The cells divided once, and the cells have divided again. So in meiosis, we have 2 cell divisions. Whereas in mitosis, the parent cell only divides once, and these 2 daughter cells are genetically identical to the parent cell. So let's have a look. Which 2 statements are true for cell division by meiosis? Okay, so 4 daughter cells are formed, and the parent cell divides twice. The DNA replicates twice? No, it doesn't. The daughter cells are genetically identical? No, they are not. Daughter cells have 2 sets of chromosomes? No, they have 1 set of chromosomes. Like I said, they are haploid cells. Endocrine glands or endocrine system. Hormones are released from endocrine glands. Each hormone travels in the bloodstream to a target organ. Label the endocrine glands. So we've got the thyroid gland in your neck, 2 adrenal glands which sit above your kidneys, and then obviously this is a female, that is ovaries. Question 4.3. So complete the table. So hormones are released in the bloodstream. They travel through the blood to other parts of the body where this hormone or these hormones target. And we call those areas target organs. So luteinizing hormone, LH, is released by the pituitary gland. And you should all know that LH causes ovulation. What does that mean? That means an egg is released from the ovaries. So LH must be targeting the ovaries or the ovary. Adrenal gland produces the hormone adrenaline. And when you have adrenaline in your bloodstream, one of the things that we notice is our heart rate increases. So that means adrenaline must be targeting the heart. Glucagon is a hormone released by the pancreas. It's released when our blood glucose levels are low, and it targets parts of your body where glycogen is stored, and it causes glycogen to be broken down into glucose. So that must be liver and muscles, liver or muscle. If you write just liver, you would still get a mark. Now we've got geranium plants. They can reproduce sexually or asexually. Suggest 2 advantages for garden centres for growing geraniums by asexual reproduction compared with sexual reproduction. So we start off with the obvious thing, only need 1 parent. And we all know that all the plants will be identical, so will produce identical plants. And then the question then says, right, what about 2 disadvantages? Now all these plants will be genetically identical, so they will all be susceptible to the the same disease if there is a particular gene that codes for a disease or if they are infected. You can also say there's no genetic variety for new colours. You can also say there's no genetic variety, which leads to weaker or unhealthy plants. So genetically identical, so will all be to same diseases. Okay, so there's no genetic variety for new colours. So remember, as customers, so if I'm out there saying B&Q trying to buy geranium plants and all of them are the same colour, it's pretty boring. So as a customer, it'd be nice, to have different colours. But obviously, with asexual reproduction, the disadvantage is that there is no genetic variety. They all look the same. Okay, question 5. So Punnett squares. This question is about inheritance. Humans have 23 pairs of chromosomes in each body cell. One pair of chromosomes determines sex. Complete figure 5. So the female has XX chromosome. The male has XY chromosome. Complete the Punnett square. So that's X, XX. And be careful. Make sure the Y is clearly written. XY. So there's a 50% chance that the offspring will be male, 50% chance that the offspring will be female. In some families, all children are the same sex. Give the reason why. So you can say that each child born has a 50% chance of being male or female. Cystic fibrosis. So let me just give you a bit of insight. So on the spec, you need to know cystic fibrosis as an inherited disorder, and you need to know polydactyly as an inherited disorder. Cystic fibrosis is caused by a recessive allele, which means you need to have 2 recessive alleles in order to have the disorder. Polydactyly is caused by a dominant allele, so you just need at least one dominant allele to get the disorder. So a man and a woman do not have cystic fibrosis. The man and the woman have a child who has cystic fibrosis. We know that's only possible if both parents are carriers or heterozygous. Both parents have to be heterozygous. Draw a Punnett square to find the probability that the next child will have cystic fibrosis. Use these symbols. So, let's draw the Punnett square. Okay, so on this top section here, let's go for the woman, the man, and like I said, both are heterozygous, so both alleles are different. So capital T, lowercase t, capital T, lowercase t. What you have in these boxes represent gametes. So obviously in a woman, that's one egg cell, possible egg cell. That's an egg cell. In a man, that's a sperm cell, that's a sperm cell. And what the Punnett square is trying to tell us is this. So if this sperm cell here that's carrying the dominant allele fertilizes this egg cell that also has the dominant allele, the fertilized egg cell would have the following combination. And so on. Always put the capital letter first. Yeah. So, it says draw a ring, and this is the bit the students always miss out for some reason. It says draw a ring around the genotype of any child with cystic fibrosis. So, as I told you earlier, cystic fibrosis is caused by having 2 recessive alleles. So, this is the child with cystic fibrosis. What is the probability? Well, it's 1 out of 4. So you can say 1 in 4, or just go for 25%. You can even say 0.25, but the easiest, what students find easier to do is having that probability as a percentage. And then we've got embryo screening. That can be done by 2 methods. Method 1, the woman becomes pregnant by sexual intercourse. After 10 weeks of pregnancy, a fine needle is inserted into the uterus. Sample of fluid is taken. DNA of the embryo is tested. The screening is done free for couples who are at risk of having a child, small risk of miscarriage. Method 2, in vitro fertilization. That's fertilization outside of the body to produce a small number of embryos. Each embryo is tested for cystic fibrosis. 1 or 2 embryos with cystic fibrosis are implanted. In 2017, the success rate for having a baby by IVF for women under 35 years was 30%. It's stressful, invasive procedure. In most UK areas, the free public health service does not provide IVF for all women asking for it. Suggest 3 reasons for choosing method 1 instead of method 2. Well, the obvious thing, it does not need IVF. Which is invasive or stressful. It also talked about that it is free. The test is free. And then whilst this test can be carried out even once the mother is pregnant. So just one advantage of method 2. So here you can say that the most obvious thing is the child will not have cystic fibrosis because each embryo is screened. Also, you can say that the parents don't have to consider termination. And the other thing you can also mention is that it's a way to have a child if couples are having fertility problems. Okay, so question 6. So scientists believe that the first life on Earth was primitive anaerobic bacteria which appeared billions of years ago. Which domain of the 3-domain classification system do these primitive anaerobic bacteria belong to? So what I'm going to do is if I just quickly do a recap. Okay, so this is looking at the classification system. So the 3-domain 6-kingdom classification system was proposed by Carl Woese, and that wasn't very long ago. Okay, so Carl Woese said that all living things can be classified into 3 groups, 3 big major groups, which he called the domains. Okay, So the domains. So he said you have got the eukaryota, basically organisms that are made up of eukaryotic cells. He came up with archaea and then bacteria. The 3 domains. I'll come back to these two in a minute. Eukaryota are your animals, your plants, your fungi, and your protists. Archaea, you can just call it archaea again, keep it nice and simple. Bacteria, you can call it just bacteria or eubacteria. And if you have a look, 1, 2, 3, 4, 5, 6. These are your 6 kingdoms. So animals, plants, fungi, protists belong to the eukaryota domain. Archaea are your primitive bacteria. They've been on Earth for a very long time and they live in extreme conditions where no other living thing or any other bacteria can survive. So these bacteria live in extreme conditions. And bacteria here are your everyday bacteria, what you have on your hand, your table, your books. Okay, these are bacteria that cause diseases. Okay, so this was proposed by Carl Woese. And then Carl Linnaeus, remember around about 1700, your Swedish botanist, he came up with the classification system where you would look at each of these kingdoms and you divide it up even further. So what I say to my students is, remember this mnemonic: kids prefer candy over fancy green salad. Just remember this mnemonic. K for kingdom, P for phylum, C for class, O for order, F for family, green for genus, and then you've got species. This classification system here was put forward by Carl Linnaeus, and we still use this system. So here's your kingdom, so animals, that would go here. Okay, you don't need to know the specific names for each, but you would then put it into a phylum, group, class, order, family. Genus for animals is Homo, species is Sapiens. Okay, you will never be asked to give the specific names here, okay, but you do need to remember this order here. So these are your kingdoms, and then you carry on classifying, okay? Then also you need to know the binomial system. So binomial, again, was proposed by Carl Linnaeus. Binomial system is a system used to name all living things. All living things have unique Latin names. So for humans is Homo sapiens. You have 2 words. First letter of the first word is capital. Second word, first letter is lowercase. I know my S doesn't look like a lowercase. It's written in italic. And when you handwrite, obviously it's difficult to write in italic, you then underline. And then the first word is the genus. The second word is the species. Okay, so that was just a quick recap. So then go back to this question. It says, which domain of the 3-domain classification system do these primitive anaerobic bacteria belong to? Primitive bacteria lived on Earth or appeared on Earth billions of years ago. I told you this here. The answer is archaea. And then you've got this graph. So ammonites were organisms that first appeared in the oceans 415 billion years ago. Ammonites disappeared in the fifth mass extinction. Draw a horizontal line on figure 6 to show the time period that ammonites existed on Earth. So they disappeared during the fifth extinction, so right there on top of this line, and they first appeared in the ocean 415 billion years ago, so be careful. 415 is in this direction, and 10, 20, so 2 squares is 10, so 3 squares would be 15. You can draw this horizontal line wherever. I've just put it up here because it's the highest point in the graph. And then you take these two, so that's 415. That's fifth mass extinction, tells me there. And then you take a ruler and you join up the 2 points there. So I can't seem to find my ruler. But you can see you draw a line from that point all the way across to that point there. And you label the line ammonites, because it tells me there, label the line ammonites. Another type of organism that existed in the oceans was called trilobites. Just 1 second, let me just see if I can find a ruler. Okay, so another type of organism that existed in the oceans was called trilobites. Trilobites existed from 544 million years ago until 278 million years ago. How many more years did ammonites exist than trilobites? So let's have a look at ammonites. So ammonites exist. So if you go here, it says ammonites were organisms that first appeared in the oceans 415 million years ago. Okay, so it's 415 million, so I'm just going to put M there. And when did they become? Ammonites disappeared in the fifth mass extinction. So that was how many years ago? So we need to find out how many years that is. So 10, 20, 30, 40, 50, 60, 70, 65. So 65 million years ago. If you put that in your calculator, you should get 350 million years. Yeah, then we do trilobites. So they appeared 544 million years ago, became extinct 278 million years ago. The difference there is 266 million years. And then you work out the difference between the two. It says how many more years. So you go 350 million years, subtract 266 million years, and that should give you 84 million years. There was an increase in the percentage of species which became extinct in the third mass extinction compared to the first mass extinction. Calculate the percentage increase. So if you look at the third mass extinction, your highest point there, read that off. So 96, that's 96. then look at the first, sorry, the first mass extinction. Read that value of 68. And the difference there is 28. Because I'm looking at percentage increase, I'll do 28, divide that by 68, multiply that by 100. That gives me 41.18. I can round that up to the nearest whole number, 41, or I can just leave it like that. So 41.18%. So, next question. Human activities affect the environment. Describe the reasons why deforestation takes place and the effects of deforestation. So, why does it take place? So, the reasons for deforestation. So, 6 marks. So, I'm going to write 3 reasons and then I'm going to write 3 effects of deforestation. So, we're clearing up rainforests for land. So clearing for land. Why? To grow more crops, because obviously the population is increasing. Land is also being cleared for mining. I can also mention land is being cleared for growing biofuels. And land is being cleared for farming, to obviously, have to rear cattle, because we need more meat, so we need more animals, we need more land to keep those animals. So those are my reasons. Effects. So I've got less trees now to take in CO2 for photosynthesis. We're burning the waste wood, which releases carbon dioxide. So we're burning wood to release CO2. We're reducing, this is leading to reduction in biodiversity, because if I'm cutting down trees, I'm getting rid of habitats, animals are dying, or animals will have to migrate to other areas. So reduces biodiversity. Habitat loss. I know I've gone beyond 3 bullet points. Habitat loss, like I've mentioned. And then you can talk about the CO2 here, which will contribute to global warming. Forests have been called the lungs of the planet. I mean, these 2 questions are really, really strange. Now, describe one way forests being called the lungs of the planet can be considered to be a correct statement. Because trees are also exchanging gases. They take in CO2 and they release oxygen. So, involve gas exchange with the atmosphere or the air. But trees don't have lungs. Describe one other way forests being called the lungs of the planet is an incorrect statement. So here you can say carbon dioxide is used, oxygen is released, whereas in the lungs it is oxygen that's used and CO2 is released. So carbon dioxide is used and oxygen is released. That's obviously in trees. Whereas in lungs, oxygen is used and CO2 is released. And that is the end of this paper. I hope you found this video useful. Hope to be back with another video very soon.